Okay, I'm going to start off just with a little bit of background, which isn't really genetics at all, just to put everything in context. And the point I want to make is that quite a lot of this talk is about blood capillaries, and blood capillaries link the arteries to the veins. And so the first diagram, if I can make it work, is simply to show that that's the way the blood goes. And a blood capillary is a pretty thin, pretty small tube, quite long obviously, and it's composed of lots of cells. Here's a cell, here's another cell, and in cross-section it looks something like this. And a cavernoma fits on a blood capillary in the way that you see there. So that should be all pretty familiar to you, I think. So here's the blood capillary in cross-section at the top here. And a normal blood capillary, that is one that doesn't form cavernoma, has, and this is another cross-section of a blood capillary down here, has what are called tight junctions between the cells. And that's indicated by this little sort of black thing here. And those stop anything leaking out of the cell. Clearly nutrients or oxygen go out through the cell, go actually through the cell from the blood to the brain, but blood platelets, red blood cells or whatever can't get out through there. And this is part of what's called the blood-brain barrier. But in a cavernoma, the cells have lost these tight junctions. And so there aren't any tight junctions between the cells here. This means the cells can move apart slightly and they can become leaky. And so red blood cells can leak out through the gaps in the cells. I'm not sure that's all the time, but that's the basis of a cavernoma. The cells here are shown enlarged. In fact, in a cavernoma, you get more cells, you get multiplication. But I shall do another talk I think in August or September, where I shall talk about the structure of cavernoma. This is just purely schematic. And the important thing about this particular slide are these tight junctions. They are very important to how the genetics works. So we get on to genetics now, and I'm going to start with human chromosomes. We have 23 pairs of chromosomes. You get one from your mother and one from your father. And this shows the 23 pairs of chromosomes. Well, it shows 22 pairs of chromosomes down to here. And then there's an X and a Y. You get one from your mother and well, an X from your mother and an X or a Y from your father. So that forms the 23rd chromosome. Now we have about 25,000 genes. So 25,000 genes, 23 pairs of chromosomes. That means you get, roughly speaking, 1,000 genes on each chromosome. Now they're different lengths, as you can see. The number 1 is the longest, and that has around about 2,000 genes on it. And number 22 is the smallest, and that has something in the region of 400 genes along it. But you have to imagine that there's a sequence of genes running along the length of the chromosome, G1, G2, G3, down to G1000. Okay, now there are 3 cavernoma genes. They're known as CCM1, CCM2, and CCM3. CCM3 is on chromosome 3, so here it is on chromosome 3 somewhere. I'm not sure exactly where, but maybe here. And CCM1 and CCM2 are on chromosome 7. Here they are over here on chromosome 7. Now I want to make the point that any gene is located in a particular place. Let's just talk about G2, some arbitrary gene at the moment. That gene can be in a normal form, which would be the case in the bulk of the population, or it can be in an abnormal form. And so if we're talking about cavernomas, can be implicated in giving rise to a cavernoma. In other words, the gene that you get in that location isn't always exactly the same. The word that we use is allele, and it means that this gene has more than one allele. It has the normal form and it has the abnormal form. And indeed, it might have more than one abnormal form. But what occurs at this location is always having the same kind of output. It's a unique gene for a particular purpose, even though sometimes it isn't there. How are we doing so far?
Good.
Hands up if you can vaguely understand me still. I can see one or two people without hands up. I'm going to take it that that's because they're shy of putting their hands up, Sonal. Okay, so what does a gene do? A gene is an instruction to give rise to a protein in the cell. So here's a normal gene, and here is the protein that it produces. And this is the gene CCM1, and I've written a red N there for normal. So that's indicating that it's a normal gene, and it gives rise to the normal protein. So CCM1 does that for a CCM1 protein, but CCM2 will do it for a CCM2 protein, and CCM3 will do it for a CCM3 protein. In other words, the CCM genes in their normal form give rise to 3 proteins. And those proteins form a complex. They combine together to act as one unit. So this is the unit which is important for Cavernoma. What happens in the abnormal situation? Then in the abnormal situation, we have the Cavernoma gene, that is, it's become abnormal, and here it is, a CCM type as opposed to the normal type. And in that case, you don't get any protein. And that's the same whether it's CCM1 or CCM2 or CCM3. And whichever of those it is, you no longer get the complex. So this complex that you get in the normal, operation of the blood capillary cell we're talking about, doesn't have that complex in the abnormal form. Now it's slightly more complicated than that, and I'm going to just show you why. Here's a person with 2 normal genes, their mother and their father. Neither of them has the familial form, and so you have the normal form of both the genes, one on chromosome from your father, one on the chromosome from your mother. And so each of those gives rise to this complex of proteins, and so the cell has got lots of these complex of proteins in them. And those proteins give rise to, they aren't the tight junction themselves, but there's a sequence of reactions that comes along here that gives rise to a tight junction. In other words, you have to have this complex to get a tight junction, and you have a normal blood capillary. If you're born with the familial form of Cavernoma, one of your genes is abnormal and the other one is normal. You never get the situation where if both the mother and the father have, for example, CCM2, you never get the situation where you have 2 abnormal genes. Those fetuses would be aborted early on. They're not viable. Now, in this situation, what happens with the complexes? And the answer is the one which has the Cavernoma abnormal gene gives rise to nothing, and the one that has the normal gene gives rise to the complex. And that is sufficient to produce enough of what happens along here to give a tight junction. And so you get a normal blood capillary. So the familial person is born, if they're born in this sort of way, with they will be born in this sort of way with one Cavernoma gene abnormal and one normal gene, they will have normal blood capillaries. In order to get a Cavernoma, both the genes on the 2 chromosomes, one from your mother and one from your father, have to be the abnormal form. And in that circumstance, neither of them produces the complex, you don't get a tight junction, and so you get a Cavernoma. So the question is, how do you go from having this down to here, or having this down to here? I'm not too good at this pen. Because unless something happens, you won't get a Cavernoma. I'm going to keep asking, Hands up if you're okay.
Yeah.
Okay, I hope that's fair to ask you. Pat, you have to tell me if I'm asking too much. So in order to understand how we go from having one of these to having both, we have to digress slightly. So second digression. And this talks about the cells in your body. You have sperm or egg, depending on your gender, and the sperm has got 23 chromosomes. The egg has 23 chromosomes, but only single ones. Only when you get an embryo do you get the situation where the cell has got the pairs of chromosomes. This over on the left-hand side are inherited. These over on the right-hand side are not inherited. And all the cells in your body that are not the sperm or the egg are called your somatic cells. That is the nerve, the muscle, kidney, liver, the blood capillary cells, which matters for this talk, are all called generically somatic cells. And you can get mutations in those somatic cells, and those mutations are not inherited. And you can get mutations at any point along the development of somebody, right through when you're still in embryo, right through to when you're a child, to when you're a grown up. And so you can get mutations in a somatic cell along the life along your life. And those are called somatic mutations. Now, a somatic mutation can be because there's an error in replication of a chromosome during cell division. It can be due to external radiation, and there's external radiation in the environment the whole time. It's at a low level. It can be from having X-rays when it's a bigger dose for a very short time, but there's external radiation all the time. It can be for toxins, and no doubt it can be for other causes. But the point I want to make here is that somatic mutation will happen on a single gene, in a single chromosome, in a single cell. There are somatic mutations going on throughout your life at quite a high frequency, but that's because you've got an awful lot of cells in your body. The probability that you will get a chromosome, a single gene on a chromosome in a single cell getting a somatic mutation are far lower. So let's go forward here. If you have the familial form of the Cavernoma, that is, you have one abnormal gene and one normal gene, you then have to have a single hit, a single somatic mutation in order to get to the possible formation of a Cavernoma. And that has to happen in one of your blood cells, in one of the capillary cells, excuse me, one of the capillary cells has to have a hit on the Cavernoma gene. It might be having hits all the way elsewhere along here. They won't in general matter. If you have a liver gene, then having a mutation in a liver gene doesn't do any harm at all. You can lose a liver cell without worrying about it. But if you have a Cavernoma gene, then you have, after a hit here, then you can get Cavernoma formed, and that's a familial form of Cavernoma. If you are a person that's going to have a sporadic Cavernoma, you start off with two normal genes. You have to have a fit in one of them to give rise to an abnormal gene, and then you have to have a hit in the second one to get an abnormal gene, and you then have what's called a sporadic Cavernoma. And this is known as the two-hit mechanism. And that isn't unique to Cavernoma. That's found in many, many situations. It was first found, I think, in breast cancer with what are known as the BRCA genes, where the two-hit mechanism is required to get the cancer potential for breast cancer. But in Cavernoma, this two-hit mechanism works in the way that I've described here. Is this still understandable?
Yeah.
You're beginning to lose me, David.
Pat, you'll have to translate for me.
Ian said you're beginning to lose him.
Right. Can you ask a question to get you back on track, Ian?
Yes, can you describe again the difference between familial and sporadic?
Right. If you are born with a abnormal gene, a familial person has got one gene from one of their parents as the abnormal one, and one from the other parent as a normal gene. If you are born without the familial form, just in the normal form, both the genes for this Cavernoma gene are normal. So that's the difference. You have a different starting position. If you are born with a genetic form of Cavernoma, this is the starting position you have. Does that make sense? If you are born without familial Cavernoma, you just have two normal genes. Okay?
Yes.
And so if you have familial Cavernoma, you only have to have one hit. It must be on this gene, on this chromosome, because then you will get two abnormal genes on the two chromosomes. And that's then means you don't get that complex of proteins forming, and so you don't get the tight junctions. If we go back to this slide, then the familial form has one complex, but when you get to the two abnormal genes, you no longer get any of this complex, so you don't get a tight junction, so you can get a Cavernoma. Does that help?
Yes, it does. It's very clear.
So here we have the two-hit mechanism, and this is the final slide you'll be pleased to hear. Somatic mutations are random events. We don't know the frequency in blood capillary cells, but it's probably an average of one mutation on one particular gene on a particular chromosome in a particular cell once every few years. It will happen on other cells at the same sort of time. But by saying it's on average of one every few years, it could only be 10 minutes, but it might be 10 years, but on average it's 1 year. Okay? Whereas if you have, if you have to have just one hit to get a familial form of Cavernoma, that can happen quite easily. It might only be every year or so on average, but nonetheless, you will get lots of familial Cavernoma. But if you have to have two hits, you'll get one hit on one of them first of all, but then to get a Cavernoma, you have to have another hit on the adjacent chromosome, the other one of the pair, on that same gene, in order to get this pair of abnormal chromosomes in that one cell. And the chances of that happening are very much less than in the familial case. This double hit is a rare event compared to the familial form. Yes? I'm not seeing many smiles.
It's just a lot to take in.
It's a lot to take in, but it's there.
It's there. And if you are recording this, this is great.
So familial Cavernoma will tend to appear at a lower age, at a younger age, because you only have to have the one hit, and the chances of getting one hit are much higher than the chances of getting two hits. Not only will they appear at a younger age, the probability of having multiple Cavernoma with familial gene, because this is one cell, but then there will be another blood cell, another blood capillary cell somewhere else, to which the same thing will be happening. So you can get Cavernoma all over the place easily with a familial form, but with sporadic, you've got to get the second hit in the particular gene onto particular chromosome in that particular cell, and that's much rarer. Now that's it.
Can I ask something? What decides where the Big Bang is going to be in your body?
Where the band is going to be?
Where the Big Bang, I was saying where the event is going to happen.
It's randomness.
Yeah.
I mean, if we go, I mean, let's take it that it's due to radiation, which it might, I mean, it often is. Radiation is just a random event. The radiation, which is in the environment, which can be from the rocks, it can be from a variety of sources. It's a low radiation, will hit randomly on any cell at any time.
Mm.
It's just purely random. And
David,
if it's,
I'm sorry.
you say?
David, I thought you were finished. I'll let you finish that, but also
I have, you say, you ask.
Sandra has a question. What if both parents have sporadic Cavernoma?
Well, when you say both parents have sporadic Cavernoma, I think what you, if you have a sporadic Cavernoma, then you're down here. So you've got a Cavernoma, and both parents have a sporadic Cavernoma, but these aren't in the germ cells, and so they're not inherited. In other words, if you get a Cavernoma, that will be because a particular a particular blood capillary cell has been mutated, had both of these formed like this, but there's no way that passes on to your children, because it's only through your sperm or egg that the children will pick up the mutation. Does that make sense?
No.
No.
Okay, good. That's fair. I mean, thank you. I'm going to go back. I'm going to go back a slide. Sorry. I'm going to go back here. If you get a sporadic Cavernoma, that is formed on a particular red blood capillary cell. Sorry, on a particular capillary cell somewhere in your brain. But the mutations, the abnormalities in the cells or in the cell that's responsible for this Cavernoma doesn't go to, it's unique to you, as it were. It doesn't go, it can't be, this can't be inherited by your children. So you have a sporadic Cavernoma because you started off life with just normal genes in that situation, and then you just happen to get 2 hits in this red, in this blood capillary cell, and so you get a Cavernoma. And your other
one of the sperm was one of the things that could carry it through to the next generation.
Forgive me. Yes,
in which case that the child would be made from that
okay.
sperm.
Let's suppose this is let's suppose this is a blood. Sorry, let us suppose this is a blood capillary cell. So here we have a blood capillary cell. And neither of these chromosomes has an abnormal gene on. You get 1 hit, here it is, you get another hit, here it is, you get a Cavernoma.
Yeah.
That stays within this cell, it doesn't go anywhere else. In order to get it inherited, you have to have a mutation in either the sperm or the egg.
Yeah.
And that's a very rare event. Does it ever happen? Yes, it must have happened at some point to somebody.
Yeah.
But the chances of it happening in a single sperm, for example, are very low. or the same in an egg. And so the chances that you have a cell that's been mutated combining with an egg are extremely low. That's a very rare event indeed. I mean, that we talk about founder mutations because there are known to be founder mutations, for example, in New Mexico, in Ashkenazi Jews, and there's a population in the southern states of America where you can trace back to the founder mutation from an individual in the 1780s or something. I mean, it's a remarkable situation. It's very rare. This is much more common, hence people with Cavernoma, but these are not inherited. I'm not, Pat.
But David, so some of the, I mean, a lot of people in Caver families have the inherited forms.
Sorry, a lot of people
in Caver families have the inherited form. One of the, you know,
yes.
one of the chromosomes is a carrier chromosome.
Indeed. So let's suppose that.
But you know, yeah. And I have two more questions when you're, just so you know, there's two pending questions.
Okay. I mean, if one of you, if somebody has a familial form of Cavernoma and has a child, there's a 50-50 chance that gene will be transmitted through to the child. And so, on average, half the children of a couple where one has a familial Cavernoma will be passed down to the next generation. And that person will then have the normal abnormal form of the gene. So that will be a familial form of a familial form of the inheritance. I'm not making myself clear. Ask more questions, Pat or somebody.
Well, I mean, it's for, you know, the two that said, I think, how about people who weren't clear about this? Do they have questions? Does anybody have questions there? Or is that clear?
No.
Is that clear now, people? Or not? That
Can I just give an example? It's about me. Mine must be sporadic because my family has been tested genetically and the genes are not found. Would that help for people to understand? When I never had, I only had one episode when I was 42, yeah, and the second one just happened now, and it's after 18 years. So my form of, I can understand now very clearly that mine might not be familial Cavernoma, but it's more sporadic because I did work with a lot of radiation, you know, in molecular biology.
So if neither of your parents had a familial gene,
yeah,
then you were born with this situation.
Yeah, sporadic. With
and
normal.
So you've had to have 2 hits and you then have a sporadic Cavernoma.
Yeah, I understand that, yeah.
But let us take a family, and we
because
know
my children, their genetic test and their MRI shows.
and so
can you hear me?
If you have the sporadic form of Cavernoma, you can't pass that on to your children.
No, I mean, that explains because none of my children, their MRI or their genetic test, it shows no sign of any of the genes. So that's good. That's why my daughter they didn't find any genetic mutation.
Yeah.
Yeah.
But
So I can't. Sorry. So that means in my case, if it's sporadic, that means it will not be passed on to other generation in our family because neither of the children got it.
If you have a sporadic Cavernoma, it can't be passed on to your children.
Yeah.
Let me see if I can explain one other thing and see if this helps. Are you still?
Can you explain about this double hit?
I'm sorry?
Can you explain about this two hits, you know?
Can I explain it again, you say?
Yeah.
Sure.
Could I just pipe in for 1 second?
Yeah.
I think what really surprised me and what flabbergasted me when I first learned about this was when I went to school, the idea of genetics and genetic illness was all about inheritance. You inherited a faulty gene and that could lead to an illness. The thing that's flabbergasted me about this hit business was that there's two things going on here. One is there's an aspect that is about an inherited abnormality, which happens with familial cavernoma, but that's not enough. There's also something that happens, there's a second thing that you need, both in the inherited form, the familial form of cavernoma, and in the sporadic form, where there's no inherited aspect whatsoever, which is a genetic mutation and thus abnormality that happens when you're, post-birth, that has nothing to do with the inheritance line. And that to me was something I had to get my head around when I first learned about it, these hits.
Right.
And then dividing that into two. So there's a familial form, an inherited form, and a non-inherited form, but then both of them have to have this hit business. Anyway, once I got my head around that, I thought I understood it a bit better, even if not 100%. So that helped me.
Let me see if I can see if this helps. If your parents both have the normal gene, then you will inherit just normal genes. And so you will have to have these somatic mutations to get a cavernoma. You get a sporadic cavernoma.
Yeah.
Let's suppose that one of your parents has a normal gene on one chromosome and the CCM gene on the other. Then the children are born with I'll stop there. If your parents, either parent has familial cavernoma, they will have in their egg or sperm one normal and one abnormal cavernoma. When an egg is formed or a sperm is formed, it takes either this one or this one from this parent, and it takes, let's suppose it's normal for the other parent, and it takes one of those and one of those from the other parent, and so 50% of the children, or there's a 50-50 chance that you will get a normal gene from this parent and a normal gene from this parent, or you will get the abnormal gene from this parent together with a normal gene from the other parent. Does that make sense, or
Yeah.
Does that make sense to everybody?
Okay.
Yeah, but but there's still the randomness there.
It's still random, absolutely.
Yeah.
I'd go back and so here's a parent with a normal and a cavernoma gene. Half their sperm get the normal, half their sperm get the abnormal, or if it's an egg, half their eggs get the normal and half their eggs get the cavernoma gene. And so whether your offspring gets a normal gene or the cavernoma gene is a 50-50 chance. And so some of your children might have to be carried on with the cavernoma gene and some might not be.
David, can I interrupt and ask a related question that's on the chat line?
Yep.
Okay, so someone says, so when you have a familial cavernoma, so, a cavernoma when one of the abnormal genes is inherited, could a researcher take any cell from your body and find the CCM mutation?
Could you just ask the question again, please?
Well, you'll see it on your chat. So, you don't have chats up. So when you have a familial cavernoma, can a researcher take any cell from your body and find a CCM mutation?
Yes, they can. Genetic test.
Yes, if you have a familial cavernoma, any cell in your body has one CCM mutation. All the cells in your body carry that mutation.
Okay, and another related question. Can you explain why a mutation or mutations in DNA in one cell, so you get that mutation in one cell, can then lead to a cavernoma forming? So a big cavernoma forming. You have this mutation, one cell, and then you get this giant cavernoma.
Well, I mean, that's a very interesting question. This is the way cavernoma form. This is the subject of another talk, and I should go into it more. But here is a normal blood capillary cell. One cell in it acquires a mutation that gives rise to both genes in that cell having the abnormal form. This is able to form a cavernoma cell. Okay, so far? And what happens is that proliferates because this cell divides, and then all the cells that originate from this one all have this double mutant form, and so they all are red. So in other words, this cell will divide, giving rise to many cells and the start of a cavernoma. This would be a cavernoma you would hardly detect. But then what happens is that it incorporates adjacent cells, and gradually you get the build-up of cells to form the cavernoma.
Right, okay. Now, another question, a bit different, but still same subject. What causes the hit? What causes the hit?
Okay, the hit is, to answer that properly, we have to talk a little bit about what a gene is. And a gene, has everybody heard of DNA? DNA, well, I go back to the sharing. Are you still happy with how this is going, everybody?
Yeah, yeah.
We're grinning.
We're grinning. Well done.
It's hilarious.
So here's our blank sheet. DNA is a long chain molecule. It's a helix. So I'm going to try and draw. Oops, Daisy. I'm going to try and draw a helix. So here's a helix. And it's actually a double helix, but I'm not going to draw two of them, but you have to imagine two of them there. This is DNA. And DNA is what's called a polymer. It's got lots of almost identical units along its length.
Mm.
So it's a chain of, they're called nucleotides, but these are little bits of the molecule which are almost the same.
Mm.
And they are the code. And they're not quite the same because there are 4 types of those nucleotides, and they're given the names G, C, T, and A.
Mm.
And in order to get, and the protein, this is an instruction to form a protein. Every 3 units here, so every triplet tells you something about the protein.
Mm.
It's what's called an amino acid in the protein. And when you get a hit, it changes one of these bases. And so instead of being, for example, G, C, T, it might, in the abnormal form, become GGT.
Mm.
And the amino acid, the protein that's formed from this one, will be one type of amino acid, and from this one will be a different one.
Mm.
And that changes the whole nature of the protein. That is sufficient to make a protein be inoperable. So mutations are when you get a hit on a particular base in a length of DNA, which then produces a change in the protein. And that change in the protein might be trivial, it might do nothing to change the operation of the protein, but it might be something to completely change the nature of the protein.
Okay, well, that's the effect of the hit. What's the cause of the hit?
The cause of the hit is ionizing radiation. Ionizing radiation acts on molecules in cells, in wherever, to change their nature. It ionizes the particular place it's hit, and that changes the nature of that molecule.
Well, that was one of your things. It could be other things too. You had a whole list, didn't you?
And they all act in that same sort of way on the bases.
But that could be random too, couldn't it, David? You said there was, an editing thing. That was your first.
That would be random as well.
Yeah.
All of these will have random effects. If you have, when I was young, we used to have X-rays on our feet that were massively, were massive doses, we now realise. And so people who had lots of X-rays on their feet tended to get problems in their feet because the radiation from the X-rays affected the cells in your feet. I'm not sure that's very helpful. But radiation acts, I'm not sure I can really, I don't understand, you're a chemist, Pat, you can explain the effect of radiation on molecules.
Ionizing radiation, but that's only one. You had this list, it's just, cells divide, and people are better biologists here, probably, I'm not a biologist, but I do know that, just we're getting mutations the whole time from all sorts of causes, and most of them don't do anything, but some of them do something.
Yes.
In the way David said. But a lot of the causes we don't know, but some of them, because cells divide, sometimes the instructions just go wrong. And that's one of the reasons we know. The other causes, who knows? Radiation we know about, and toxins. People can, if anybody knows any others, pipe in, but that's the question of all questions about causes of cavernoma, isn't it? That we as far as we just don't know where if you take that step back and ask that question in a different way, what are the causes? Don't know.
The thing I found totally surprising when I learned about it was that here am I sitting in my nice room at home, and there is somewhere in my body the whole time, every second, there's a cell, a gene on a cell, a gene on a chromosome in a cell getting mutated.
Yeah.
Most of the time, it's totally irrelevant, either because I don't mind losing a liver cell, or because the effect of that on that particular gene doesn't actually affect the protein's function. And so the chances of it being serious are really quite low, because it's got to be a hit on a particular cell that matters to your body. And it so happens that your blood capillaries are that, but you only get a mutation on average once every year or so in any particular Cavernoma gene. If you are like I am, I imagine, a person with normal genes throughout my body, the chances of getting a double hit are rather low. If you're Pat and you're unfortunate, she will have had a hit in one of her genes, in one of her blood capillary cells in her right temporal lobe, and sometimes she happened to have a second hit on the adjacent chromosome in that same cell that then allowed her to form a Cavernoma. Whereas Tom Jones, who's got a familial form of Cavernoma, has got several children. Some of those have picked up the familial abnormal gene in their blood capillary cells. All their blood capillary cells have one abnormal gene, and so they only have to have a single hit, and the chances are that that will happen in their lifetime. It won't necessarily, but it might well do, and it might well do many times. And so we know of people in Cavernoma Alliance who have familial Cavernoma, I mean, I know of one person who has over 100 different cavernoma in their body, in their brain. Cavernoma dotted. I mean, you look at the MRI of their brain; it's full of black dots.